Hi, everyone. Good morning.
Hi, all. Good morning.
Yeah, we're starting in a few minutes just to give a couple of minutes so that other people can join. We're going to start at exactly 11:02 to have more people join. Hi, Abel, over to you now.
All right, I'm good. I'm good here now. So we should be set. Yeah.
Yes.
Thank you. All right. I think we are now on 11:02, so we'll go ahead and get started. Rob, let me share my slides here. Just to show us our agenda today. Yeah, good morning, everyone. I'm your host today, working in coordination with our project manager, Chika. And my name's Abel Ekiri. I'm a veterinary epidemiologist based here at the University of Surrey within the School of Veterinary Medicine. On the slide here, we have our agenda for this 1 hour session. We'll start off with a brief intro from myself, followed by our speaker, who will take around 30 minutes. And we will then finalize with a Q&A, and then we'll wrap up at the end of that. Trying to progress my slides. I don't see them progressing. Let me see if I can use something else here. Perhaps that will do. Yeah, briefly about our housekeeping. Remember to introduce yourself in the chat and also mute your mic. If you have questions, we request that you raise your hand during the session or during the Q&A. Alternatively, you can use the chat function. The recording is already in session, so the webinar will be recorded and will be made available via the network page. I'll briefly touch or provide a bit of background to the network. The People AMR network brings together researchers from several disciplines working on AMR with the intent of identifying better ways to develop, evaluate, but also implement interventions that promote prudent antimicrobial use within communities. We focus on people specifically because it is the behavior of individuals, but also groups that determine antibiotic use. Struggling to progress my slides here. For some reason, let me see. That's not helping. That's better. Okay. Here is our team. As you can read on this slide, we have Sarah at the very top, who's the network lead from the University of Oxford. We have Gail, who's a co-lead as well from the University of Oxford. And we have Sue at the University of Reading. We have myself from the University of Surrey. And we have Wendy from the University of Manchester, and our project manager, Chika. Here is a quick outline of what our audience is. The network in general targets communities, and by communities, we're referring to individuals who use antimicrobials. And the communities include prescribers, but also the actual users themselves. And when you look at the prescribers specifically, these cut across the different health professions, that is veterinarians, dentists, pharmacists, as well as GPs. And for the users, these cut across the animal health sector, but also people in general, and obviously looking again here at different sectors if you break them down. Okay. Our aim is, as we've stated here on this slide, we're interested in developing interventions with people and prescribers that best support sustained behavior change. And how we are doing this, we are bringing together expertise from various disciplines, but also utilizing various approaches, some of which are listed on this slide. And we bring all this expertise and disciplines together to develop interventions at the community level, that is factoring in the prescribers, but also the actual end users, as we saw on that last slide. The activities are several. They range from building networks as well as collaborations within the UK, but also outside the UK. Knowledge exchange as well as dissemination activities. We have several activities related to capacity building for EMS AMCLs, that is workshops, placements, but also webinars such as this that we are having today. And lastly, joint funding applications that we collaborate on with partners within the network, but also other relevant partners outside the network. I would like to introduce our speaker today. I have a brief bio that I will read out. Professor Roberto is a colleague here at the University of Surrey and is a veterinary microbiologist with a particular interest in zoonotic bacterial pathogens of livestock, poultry, and companion animals. His current research focuses on understanding the pathology of zoonotic pathogens, antimicrobial resistance, and the development of novel intervention strategies, including diagnostics, vaccines, prebiotics, probiotics, and postbiotics. Roberto has published over 240 peer-reviewed publications in the area of veterinary microbiology and is a member of a number of scientific advisory committees. Roberto is currently a professor of veterinary microbiology and pathology in the School of Veterinary Medicine and is also the interim pro-vice chancellor and executive dean for the Faculty of Health and Medical Sciences at the University of Surrey. Roberto's presentation will discuss the development and validation of novel farm intervention strategies to combat AMR, including the use of vaccines, post-care diagnostics, novel antimicrobials, probiotics, and postbiotics. And he will also explore how behavior changes in farmers and vets may be harnessed to increase the use of intervention strategies. With that, I would like to hand over to Roberto.
Thank you, Abel. That's fantastic. Thank you. And it's a pleasure to be here today to be able to speak to this group. And I look forward to having a discussion at the end of the presentation. So just let me share my slides. Okay, hopefully everybody can see the slides now and they're on full screen. So as Abel has kindly introduced me, thank you very much. I'm going to speak today about novel intervention strategies to combat AMR at the farm level. And I thought I would start by just really emphasising that our behaviour, whether it's direct or indirect, can influence AMR. And of course, over the last few years, we've seen a significant change during COVID in pet ownership, not only the numbers, but also the types, and some of which perhaps are more normally associated as being farm animals. We're seeing more and more poultry being kept as pets. They're a popular pet now. And as you can see here, it's we're being urged not to perhaps kiss those pets, particularly those ones that might have zoonotic pathogens or harbour particular types of antimicrobial resistance. Alongside that, we've seen an increase in household domestic pets too. It's been really good from a perspective of the responsible use of antimicrobials in animals over the last few years to see the huge efforts that have been made to reduce the sales and use of antimicrobials. And the most recent VARS report, which is just depicted here, it gives good illustration of how that work has progressed over the years. We've also had new legislation come in from the EU and others, along with a number of associations such as RUMA, who really championed helping to reduce antimicrobial use in animals, be it companion animals or indeed livestock or poultry. And that's really pushed antimicrobial stewardship. And we've had a huge amount of work from the British Veterinary Association, from articles describing different types of antibiotic stewardship or antimicrobial stewardship, right the way through to simple guidance documents which can be put on a wall in a veterinary practice to really give clear definition around responsible use of antimicrobials in practice. And the VARS report from the Veterinary Medicines Directorate, as I mentioned before, illustrates that significant reduction over years of antibiotic sales and usage. And on the left-hand side, because we're concentrating particularly on livestock and poultry today, you can see the really huge efforts that have been made over that time in the reduction of antimicrobial usage in food-producing animals over the years. Obviously, there are fluctuations from year to year, but overall, the trend is very, very, very positive. So why then do we need alternatives to antibiotics or indeed antimicrobials? Well, the development of antimicrobial resistance is right at the top of our minds. Organic production systems where antimicrobials are not generally used, cost of the antibiotic treatments, consumer market pressures, alternatives to growth promoters where they were used previously. But very importantly, there are few new antibiotics in the pipeline. So even if they do come through, we need to think about what are the other alternatives that could be used, either as complete alternatives or to augment existing antimicrobials or repurpose. Positive thinking around these alternatives, many of them can be used as feed additives that can be added to the feed or indeed water. Alongside that, we need to think about how do we influence farmer and vet behaviours in order that many of these different types of interventions can be successfully adopted. And that can really relate to economic drivers and constraints, whether we have knowledge and education, identifying what the social norms might be, perceived risk and experience, time and labour burden. You know, is it a vaccine that needs to be given by injection? Could it be put in the water? And how information is communicated. You know, what do we know about those interventions? What is the science behind them? How is that communicated to the end user? So with that in mind, what are we looking for in a good intervention? Well, we needs to be cost effective, needs to be evidence-based, easy to administer. As we're thinking, some of farms I visit have, you know, huge numbers of poultry, for instance, on those farms, and, you know, tens of thousands in each shed, and you have to think about how that's going to be administered effectively. So perhaps in the water again, or feed or spray. Effective against the target pathogens, stable, so it can be used in different environments, specific, and sustainable, really, really important. So what might those alternatives be? Well, there are many, and a long list here, which I'm not going to read out, because you can all see the list there. And on the right-hand side, some of the papers that we've generated from the work that we've undertaken in my research group. And I should say, and I will later on, all of the work that I'm presenting here has been done by a huge group of individuals within my own research group, but also a wider network of collaborators. But as you can see here, there are some novel things, natural plant extracts and phage therapy through to fecal transplants. But there's also things that we're all familiar with, vaccines, probiotics. And these are genuinely really good alternatives to antimicrobials. But where do we start? I think we need to remember that when we're treating with antimicrobials, we're not necessarily just having an effect on the target organism. And this is something that my group has really taken on board, and really think about the holistic influence of an antimicrobial alternative to the normal flora. So our starting point is always, can we understand the microbial diversity? And when I talk about microbial diversity, I'm a bit biased because I'm a bacteriologist, so I think about the bacteria a lot. But of recent, we've been working with lots of colleagues to understand what happens when we intervene or there is an infection. What happens to, for instance, viruses, parasites, fungi? And just at the bottom there, illustration of a couple of papers where we've been looking at the influence of avian influenza on the bacterial microflora. We also want to think about how that antibiotic treatment influences overall the microflora and performance. And here I'm using an equine example because it's one of our recent publications where we've shown that antibiotic treatment in the early life of a foal has a profound effect later on in life. And we measured here performance from a perspective of racing, but we also looked at number of days that the horses were unable to train, and health incidents, whether they were respiratory, diarrheal, orthopedic or other. And we showed that in those first few weeks, indeed in the first few months of life of a horse, the type of antibiotic used, how it's administered, can have a really big effect on the microflora for a significant amount of time. So similarly, we feel this would also happen with livestock, and we know that in many cases, and in this case we were looking at a treatment of Brachyspira infection in poultry, and we know that treatment with antibiotics triggers dysbiosis. So we've got a change in that microflora. Not only are we treating a particular infection, but we result in a fluctuation in the normal flora, and therefore those animals might be more susceptible to other infections. We also need to think about how that microflora influences pathogen carriage. And here we looked at a number of farms where we were looking at Campylobacter carriage. And we firstly showed that there was a significant difference between one farm and another in the microflora and related to the Campylobacter carriage. But also it was interesting to note that pathogens influence the gut microflora, even if they don't cause clinical disease. So we've got changes with the types of Campylobacter and numbers of Campylobacter in those different farms, and they are influencing the microflora. So not necessarily having more pathogen means that you've got a better flora or vice versa. Within those infected farms as well, and this is looking at different animals, specifically pigs here and Salmonella rather than Campylobacter, it's also important to be able to identify those animals which are higher risk when we're thinking about interventions. So not only the microflora that's present, but are some animals what we call super shedders? And how do we control those organisms and the antimicrobial resistance associated with them when we have numbers of super shedders within a herd or a flock? So these are animals that persistently shed high numbers of a particular pathogen. And here we were looking at Salmonella in whether they were low, high or medium shedders. So on to some of the interventions that we might think about. And I wanted to start with one area which I've done a lot of work in, and that's Brachyspira in poultry. This is a bacterial spirochete infection, which affects poultry, predominantly layers. And there are a limited number of antimicrobials available licensed to treat this infection. It can easily come back as well, so that you get various bouts of the infection, so repeated treatment may be required, and it has a significant influence on productivity, number of eggs, but also be significant welfare issue for poultry. So we embarked on a study to really try and understand what could we use as an alternative. And here we used probiotic, Lactobacillus reuteri, and we were able to show by producing our probiotic in the laboratory, but also thinking about how we could add it just to either as an oral dose or in the water to poultry. We were able to show here in the graph you see at the top here, without probiotic, it's a normal level of shedding of Brachyspira bacteria in these poultry. When we gave a dose of the probiotic, we were able to reduce the blue line, the level of shedding of that bacterium. And we were also able to look at fecal moisture content, because this is really important from a perspective of welfare. If the environment becomes very wet, it becomes the birds will suffer from other conditions related to having a wet environment. And we showed that with the probiotic, we had less moisture in the feces there. So by just providing the birds with a probiotic in the water, we were able to significantly reduce the level of clinical signs and ensure that the number of bugs being shed was reduced. And we did a lot of in vitro work prior to this study, which demonstrated that actually in this case, the probiotics were actually able to antagonize the attachment of the Brachyspira to the gut. And so it was actually a physical as well as a partially chemical effect. In a similar study, we looked at Salmonella and prebiotics. So unlike the probiotics, which are live bacteria, which we added to the water, here we look at a prebiotic, in this particular case, galacto-oligosaccharide, which acts as a food for the good bacteria. And on the right-hand side, you can see, well, left-hand side, some of the publications for it. Right-hand side, you can see that we were able to reduce from the red to the green highly significantly the level of Salmonella in the various tissues. And also at the bottom, the left-hand side shows the pathology associated with Salmonella. On the right-hand side is what we saw when we were able to have it controlled. So we also wanted to look at the effect of lactobacillus on association of the Salmonella to porcine in vitro organ culture tissue, and in this study we looked at whether we could use our lactobacillus to reduce our salmonellas adhering directly to tissue in the laboratory, and published works are on the right hand side, but you can see in the graph at the bottom that actually what we saw here was our salmonella on the left hand side, and then different types of lactobacillus, but also adjusted for pH. So this was pH independent, because we know that pH will have an effect, but irrespective of the pH, we were able to reduce the levels of adhesion of these organisms to tissue explants. So a really good option there, and that allowed us to take it one step further using our in vitro gut models. We showed that where we were able to add lactobacillus plus a prebiotic, so a symbiotic where we've got both together, we were able to reduce the numbers of salmonella, but we were also able to increase the numbers of good bacteria, so the lactobacilli within the system. And that took us to an in vivo study. And if you focus on the black bars where we've got specifically showing that we've got the lactobacillus plantarum plus the probiotic, you can see that this depicts the levels of salmonella. The solid black bars are generally lower all across the study, showing that when we had a symbiotic, so lactobacillus plus prebiotic, we were able to reduce the numbers of salmonella being shed in the feces. So one other area which we've been looking at more recently, which is very novel, is postbiotics, and these are the byproducts produced by probiotics. And a common one is butyrate, which we know helps to keep enterocytes, so the cells in the gut, healthy. But actually what we were looking at here was, could we use the probiotics to prime the immune system in a way that would help to combat infections? So we did some literature reviewing and wrote this review here, but we also have recently undertaken a study where we were looking at priming avian macrophage cell lines, in particular in response to a number of different pathogens. The focus was avian pathogenic E. coli, but we did look at Salmonella, Pseudomonas, and staph as well within those models. And what we were able to show was that when we were able to prime those cells with butyrate, we were able to elicit a better response. So increases phagocytosis of those pathogens that are put there. So by using probiotics, we can prime the immune system so it's better able to combat infections. So I wanted to change focus a little bit to vaccines, and also think about how they've been used, because we don't always think of them as alternatives to antimicrobials, but they are very good vaccines and alternatives. And here we're just looking at one in particular for E. coli, which I've been heavily involved in the studies around. We need to take that in context that actually, if we think about global livestock vaccine sales versus antibiotic sales, there is a correlation there. So if we're using more vaccines, we can reduce the amount of antibiotics or antimicrobials that are being used, because those animals are protected from infections, so are less likely to get clinical disease and less likely to require treatment. So number of studies have demonstrated that the use of vaccines in animals can lead to significant reductions in antibiotic use, alongside providing health, welfare, and productivity benefits as well, not just the reduction in the antimicrobial use. And those vaccines are available. We know they are for some of those pathogens of foodborne zoonoses, as well as causing clinical disease in animals, both in livestock and companion animals. We also should consider that autogenous vaccines are used. They might not be licensed vaccines that are sold commercially, but they do have a place. And in some cases, we can think about, can vaccines be produced? And these are vaccines produced for a specific farm on the request of the veterinary surgeon through the Veterinary Medicines Directorate, and licensed for use just on that particular farm. And it's just an example here of a study, on-farm study that I've been involved in with Campylobacter. We have a lot of new technologies coming into, and more recently we've been using reverse vaccinology and artificial intelligence to think about how we design vaccines. And here is an example of a vaccine for Brachyspira pilosicoli, or the design of, but this could be applied to any pathogen, really, as long as we've got the starting sequence material and we're able to use those algorithms to look at potential targets. So really, we're in a very good position to think about how we push forward some of the boundaries of science where we've not been able to produce vaccines before, particularly for things like Brachyspira, spirochetes, which are difficult to manipulate in the laboratory. And perhaps thinking ahead of what else we could be doing, I was involved many years ago in a project where the ultimate aim was to produce antibodies in plants, so plantibodies. And in this particular study, we looked at antibodies to E. coli, and we were able to generate antibodies, which we would then go on to express in grass. Ultimately, we didn't make it to do it in grass, we did it in other species, so that actually you could passively immunize animals. And the potential here is huge from a perspective of being able to vaccinate against multiple pathogens. But there are obviously huge implications of getting that antibody response to work, and how can you do it from grass into the animal and it not being in some way inactivated. But what we did show in the laboratory, at the top hand side, you can see colonies of E. coli with the arrows, and when we used our recombinant antibodies, we were able to stop those E. coli's adhering to the tissue. One of the main things here was thinking about GMOs. These are obviously GMO, and how would we ensure that they could be used in the environment. Of course, now we do have GMO vaccines, and the vaccines, I've talked about previously, some of those are live attenuated genetically modified vaccines, which are used in food producing animals. What are the other alternatives? Well, there are many, and we've worked on quite a lot, and I don't have time today to talk about all of them, but other potential things that could be used quite easily. And I'm not suggesting that we have tea for our farm animals, but just as an illustration of where some of these compounds might come from. There are many compounds in plant-derived that are plant-derived, which have excellent antimicrobial activity. And we know that on farm, many phytotype chemicals are used, and they use successfully to help augment infections and treat infections rather. And I think, or support, I suppose, more accurately support the microflora in a way that it's less susceptible to infections rather than treating them. And these are not medicinal products, they're additives that are being put into the feed or water. So another is bacteriophage therapy, and we can think about how they use these viruses, which can be used to treat infections, and specifically we're doing some work around some of the foodborne pathogens, and there's some great studies being undertaken at the moment. These are easy to use, could be administered again in feed or water, and can be very specific for particular pathogens. But I suppose one thing that we need to think back to is making sure we know what those infections are before we think about treating them or enhancing the animal's resilience to those particular infections. And I think one way which we can do that is thinking about better diagnostics. And my group's been working in this area for some time, in different animal species and different pathogens. And we are at a stage now where we we do have tests which can give us results, certainly in the laboratory, in a very short period of time, in minutes. And some of these tests are now being developed for on-farm use, so that they can be used in the back of a vet's car. And we need to think about the adoption of these tests, both by vets, but also could some of these tests be undertaken on farm by farmers as well, potentially, particularly if we're looking at thinking about surveillance and where those pathogens might be. We need to think about the reporting lines. How do the results get back to the right place? Make sure they're in databases. But these tests allow us to really think about identifying the pathogen, but at the same time, knowing what the antimicrobial resistance is, whilst the vet is still on that farm. And that means that it's much easier to think about which antimicrobial should be prescribed for the treatment of that infection, so that there isn't an antimicrobial treatment that's less effective. And in that way, we can reduce the potential for antimicrobial resistance development. Other areas that we've looked at, and this has predominantly been focused on the small animal side, but there's no reason why this particular approach can't be used at the farm level, and we're looking at this at the moment. And in particular, thinking about studies which we look at from a perspective of understanding the risk of infections within a particular area. And thinking, using here virtual reality of, for instance, a vet practice or a farm to identify those hotspots. So where are the infection hotspots versus the less likely areas where bugs might be, so that we could use this, one, to train vets, nurses, farmers, to understand where the risks are, but also then use it to reduce the potential for transmission of antimicrobial resistance and indeed the pathogens across different areas. With the advent of artificial intelligence, we also need to think about how do we improve antibiotic stewardship and how can we use it. And some of the studies that we've been involved in more recently have looked at how do we bring together all of the different data sets to think about how do we predict and look at antibiotic resistance on farms. And one study which we've recently published, we looked at specifically how do we take the metadata from that farm, so antibiotic usage and sows, the number of infections that have occurred, the organisms that were isolated, and the resistance profiles, and put those all together and use artificial intelligence algorithms to help predict what are the likely risks of same or different types of antimicrobial resistance occurring on that particular farm. And that can be really powerful from a perspective of understanding which antimicrobials can or can't be used on that farm, and are less likely to result in issues further down the line. Just a little bit more on alternatives and how we might augment antimicrobials. We did a study a little while ago where we looked at a particular compound which we produced from carbon, originally metal derivative, and it was able to produce here what we call photocorms, and these are ruthenium-derived compounds. And here we were able to think about how we might repurpose an antimicrobial. And we took a particular antimicrobial and we were able to put our photocorms with that. And we were able to take a situation where normally that antimicrobial wouldn't work anymore. There was resistance to it. But with the addition of our novel compound, it was augmented in a way that it was repurposed. And therefore, we should also be thinking about how do we augment our existing antimicrobials in a way that we can avoid further resistance, but they can still be used. Just wanted to really finish off with a few slides just to say a little bit more about other projects that we're involved in. We are involved in some zinc and copper related projects, particularly looking at the influence of these compounds on antimicrobial resistance. First study, top one here, is looking at how copper in the diet and how it influences the microflora, but also how it influences particular pathogens, in this case salmonella. And the second study, and we've had follow-on studies to this, is looking at the implications of the ban of zinc supplementation in pigs on AMR in particular. And I wanted just to give a plug for some of our models as well, and really just say that, of course, this study and talk was really focused on thinking about how do we get things to the farm level. But before we do that, we need to think about what we do in the laboratory. And we've been busy in our laboratory producing a number of different types of in vitro gut models to understand how antimicrobial resistance emerges, how it's transmitted, what is the influence of antimicrobials or other treatments on the microflora and antimicrobial resistance, and how can we use those interventions. And so we're able to simulate the gut of a number of animal species in our laboratory before we get to the farm level. And in a recent study, we looked at particularly extended spectrum beta-lactamase resistance and how it was transferred not only from one pathogen to another, but also to the normal microflora. We want to stop that spread as well of our pathogens, and we have an ongoing project at the moment with our collaborators listed here, specifically looking at novel interventions for how we might actually stop the spread in animals. So intervene in that spread process, how, for instance, how plasmids with antimicrobial resistance might spread from one bacterium to another. So we've got a BBSRC project which has come to an end now that has been focused specifically on that. And we mustn't forget the environment, whether that's thinking about those microbiomes in the animals or surroundings, or the influence of things like trace elements on antimicrobial resistance in the environment, whether they're in the soil, whether they're in the manure, whether they're in the water. So in summary, influencing farmer and vet behaviours can have a positive influence on the uptake of interventions. I think further research on novel alternatives, as I've demonstrated, probiotics and others to antibiotics is required. Potentially rapid patient-side diagnostics could help reduce AMR by enhancing pragmatic prescribing, as could the use of virtual reality to enhance stewardship. We now have genome sequencing and AI at our fingertips, and we should utilize them to best effect to reduce AMR. And understanding the microflora of those animals and the environment can also help us to combat antimicrobial resistance and develop alternatives to antibiotics. So as I'm sure you can see, a multidisciplinary approach is most certainly required. And I just wanted to finish off by just highlighting the European Partnership on Animal Health and Welfare, which I'm involved in a number of projects there that focus particularly on alternatives to antimicrobials. This is a huge European partnership. Please do visit the webpage there. There is an open call at the moment. And also a plug for our European College of Veterinary Microbiology Conference for 2026, which will be held at the University of Surrey in September. So there'll be sessions focused on bacterial pathogens and looking at alternatives and indeed antimicrobial resistance. So with that, I'd just like to acknowledge the many colleagues who have been involved, so many, too many to list here, but thank you to everybody that's been involved, but also to all of the collaborating institutes and funders that have been involved in the work that I presented today. So thank you very much, and I'm very happy to take any questions.
Oh, thanks a lot, Roberto for very insightful and broad sweeping presentations on so many alternatives. I think there are a number of questions in the chat. If you can, unmute yourself if you have a question. Raise your hand and remember to unmute yourself. But as we wait, perhaps we can take on some of the questions or comments in the chat here. There is a very fast one here from Dominic. I don't know, Dominic, if you want to ask that verbally or if you want me to read that out to Roberto.
I'm fine to, yeah, so it's are evidence-based and effective the same thing? So, well, hopefully the evidence is there to show that those are effective from a perspective of when a study's been undertaken. But I suppose what I was thinking is, you know, we have to ensure that we've got the evidence that something is likely to be effective. But actually, when it's used on farm in different situations, do we know that it's effective from that perspective of, you know, have we got other infections present? Is it being used at different times? For instance, if it was a pre or probiotic. So I think there are differences there from knowing that we've got an evidence base, but actually, is it effective in the field?
Yeah, and there is an additional comment at the very bottom, Rob from Dominic. Yes, I can read it out.
Yeah.
It says clearly lots of things may be technically effective. That's whether from the lab or farm, and some may be cost effective. But could you hazard a list of certain things that are behaviorally feasible on farm? And I expect a magic diagnostic may be number one, if it existed and notwithstanding the reservations, but what else in the descending order? And I think Dominic is on the move. Maybe that's why he's not able to
no
pitch
problem.
in.
Well, no, good question. Yeah, absolutely. I think if we had diagnostics on the farm that could be used, that could be taken up by vets and farmers, I think that would be my number one. Allow us to know what the bugs are, and certainly the resistance is there. But I suppose just other things that we'd need to think about is, you know, how those farmers, for example, could use a particular intervention on the farm. So, you know, how do we, I suppose, influence from a perspective that something can be added to a water tank, can be added to feed? What is the benefit of treating for a particular infection, particularly where we have a situation where there might not be clinical disease? And we know of many examples where we look at, for instance, foodborne pathogens, where there aren't always clinical disease in the animals, but the benefit is a public health benefit. So that influence of how do we do that? And we know that there's been, you know, huge success in things like Salmonella, where the industry were behind this particularly, and thinking about how industry were involved, and it becomes the norm that vaccination happens and that you know, eggs have a Lion brand on them. We have to think of those areas where we can particularly do that. I suppose the other side is really influencing the education side, and how we help farmers who are interested in the health and welfare of their animals and the productivity, and understanding how these different interventions work. But how do we bring that knowledge to them in a way that actually is firstly understandable, but secondly translatable to use on the farm, and understand why it can be effective for their particular farm? So I think there's a big part that we need to work on in the communication part and our interaction. There's massive amount of work already been done, but I think there's more that can be done in those areas. Okay, do we want to look at some of the other questions?
Yeah, I think there is Christo has his hand up, as well as a comment. Christos, if you want to come up.
Hi, hello. Hi, Robert. Hi, Abel. Thank you very much for today's presentation. Really, really interesting. So my question is if the prebiotics and probiotics are cost effective to be applied on a farm, and if they have an effect on the final product, if this is egg, milk, meat, and finally, if they need to be applied in a routine basis in daily life, or in specific cases whenever it's needed.
Yeah, really good question. So, these are, as I said before, not really treatments. They're rather, they're additives that have been added. So the first thing in answer to your cost-effective one is it depends how they're produced and how they're administered. So in our study, one of the things that we looked at was, could we produce, and this is when the multidisciplinary part comes through, could we produce essentially what is a large bath bomb, which could be added to the water tank, which dissolves at a rate which ensures that the amount of bacteria going released into the water system is the same over a number of days, so that the birds or pigs or whatever it might be get the same number. So if you're able to produce probiotics in a stable way that could be added like that, they could be very cost effective, because you're literally just adding something to a water tank and it's dissolving. They do need to be administered, in my experience, continuously. And we know that generally with most probiotics that you need to keep the numbers high. And in our studies, what we showed that the efficacy was best where they were given continuously in the water. We have shown with other interventions, such as prebiotics, actually that single doses can have an effect. And we know some probiotics can as well. But the best effect seemed to be where we were giving them over a period of time as well. Now, to your question about the end product, I think, there's always a possibility that when you're changing the microflora, you may change, for instance, the quality or taste. So that is something that needs to be considered. In our studies in the laboratory, from a perspective of the texture, the look of it, the visually, etc., there was no change. But of course, for each product that would need to be looked at, and there may be certain products which would taint the taste potentially.
Yeah, thanks, Rob. Gail, you have a question as well?
Thanks very much for the talk. It was really interesting, and there's a huge range of stuff you're exploring simultaneously, it seems like. And I was reflecting that perhaps the pace of innovation or the pace of discovery in animals could be faster than in humans, just because of all of the additional steps we'd need to go through before we try some of these things in humans. And if you were going to nominate a couple of good bets for something we could try and move into humans from things that have worked in animals, where would you start?
Yeah, well, certainly some of the diagnostics, again, I think they would be good things to look at. I do think that some of those, obviously probiotics are used in humans, but I think we should be thinking about how do we use those probiotics perhaps as vehicles for vaccines. And this is work that is going on in many groups. So that will be another thing that I think we could look at from a perspective of how do we augment the normal flora. We know that by taking probiotics on a regular basis, they can have beneficial effects, but actually could we at the same time provide some passive immunization as well? So that will be certainly one area. I think perhaps some of the phytochemical side of things and looking at the augmentation of antimicrobials would be an interesting one. So where we looked at the photochorms, the ruthenium-derived compounds, I think there are quite a lot of antimicrobials in the human sector, which perhaps could be still last resort to antimicrobials, but it would be good to know that you could augment them with another compound, and that might mean using them at different concentrations, might mean using them for shorter periods of time as well.
Yeah, can I follow up question? So we're looking at the moment at a product which you effectively squirt dead bacteria under the tongue, and it's got some evidence that it prevents UTI, but there's skepticism about how it works. And I think some of the insights, again, it's quite hard to biopsy a person or scrape bits off a person, whereas you can do that with an animal. So some of the insights you're generating could help sort of explain, I think, why these types of approaches might work. Yeah.
Absolutely, and I think you make a very valid point. One of the issues with many of the alternative products, perhaps not vaccines, but novel vaccines, it could be, is that understanding the mechanism of action and being able to again communicate that to the farming and veterinary communities from a perspective of, you know, what is the action of a probiotic, you know, and we know that dead organisms can stimulate the immune system, they can influence other factors as well. So, but actually getting that information in a package that is easily digestible and puts forward a compelling case for that particular product, or may it be, is more difficult, I think, at the moment. And we don't have the full picture for many of these, because it is very complex. You know, we understand that maybe 4 or 5 things that probiotics might do, but they may be doing other things as well, which we're not aware of.
It's interesting. Could we take one more question? I think there is a comment, Rob, in the chat window from Kristen. Kristen, are you still on? If you wanted to comment on that.
Yes, I'm here. No, I was just highlighting some of the work we've done because Rob was talking about, as he was just talking about, uptake of diagnostics. So we did a big ESRC funded project a few years ago looking at diagnostics in the livestock arena and how vets and farmers approach those. And, you know, as you've been discussing, it's just not always so straightforward. Vets felt a little like, you know, in the UK, the term diagnosis is a protected term that's reserved for vets. And they felt like on-farm pen-side diagnostics were taking away from something that was specifically their job. So there was concern about that. And just I guess, a nod to the behavioral side that Rob already mentioned, that has that comes into play as well.
Thank you. Yeah, I think it's really important.
Go ahead, Rob.
No, I was just saying, I think it's really important to think about the behavioural side and really understand how that fits in from the perspective. I think it is an area that's more neglected, and as we develop more and more alternatives, we need to think about how that's communicated and the behaviour of those that are going to be using those particular alternatives, but also the clinicians involved in the diagnosis and the prescribing when they're antimicrobials.
Yeah, and taking it a step further there, Rob, as well as the acceptance of the animal owners, or if you're talking livestock farmers, or if you're talking companion animals, because the doctors, or in this case, the vets may say yes, but the animal owners may not be on board. So a number of aspects there definitely to explore. I think this brings us to the end, but before we wrap up, Chika has 1 or 2 slides, as well as an announcement that she would like to put across. Let me see if I can share that slide, Chika. Chika, I think I'm sharing that slide if you wanted to.
Yeah, just to let everybody know that we have an upcoming webinar on the 20th of April by 11 to 12. It will be 2 speakers from Kosovo. So we will be very grateful if you can join as well. They'll be handling an antimicrobial resistance stewardship in primary healthcare from experience of research that they did in Kosovo. So we'll be grateful if you can join, and thank you so much for joining for this session as well. And then we have the QR code here. So if you're yet to be a member and you joined by a link shared by your friend, you can scan and then sign up and receive updates from us. Thank you.
Yeah, and thanks again to our speaker this webinar, Roberto. Much appreciated. Lots of thoughts shared here, but also so many gaps highlighted. I was just reflecting on how to take this to the farmer. How do we take this to the vets, assuming we have all these alternatives working and effective? and also the questions raised by Christos and Dominic about the cost effectiveness.
Absolutely.
Yeah, so
Yeah, well,
lots of thoughts on us. Yeah.
Yeah, well, thank you very much for the invite, and thank you for the interaction and questions. I really enjoyed giving the presentation, and I'm happy to follow up if anybody's interested in further discussions. But thank you very much.
All right. Have a good afternoon, everyone.
Bye, everybody.
Bye bye. Bye bye.